Good morning. My name is Tim Hallstrom. I'm the director of the MGL. It's my pleasure to introduce Dr. Yun Yao, who runs the day-to-day operations of the Mouse Genetics Laboratory. He has great credentials, worked with Dr. Chimenti at Jackson Lab and at Cold Spring Harbor, running the transgenic facility. And he's managed to stay abreast of all the new cutting-edge CRISPR-Cas technology. So it's my pleasure to introduce Dr. Yao. He's going to give a little bit of a shorter talk, and we'll leave some time for questions at the end. So if you have any questions, think those up, be ready to ask them at the end, and we'll leave time for that. So thank you. Yun?
So my name is Yang Yu, and I'm the coordinator of the Mouse Genetic Lab. And today I will introducing some of our servers provide to the research community here. So this is a screenshot from one of the website, the servers we provide. In this column, you see the servers. Now we know it's a CRISPR, and the traditionally, we use DNA, and the second one is the ESL, and also we do the sperm cryo, IVF, and we also do the recover mice from frozen embryo and other special servers. So I will basically introducing all these categories, and if you have any question, feel free to ask. Most people probably interested in the CRISPR approach to generate transgenic mice. So why CRISPR? CRISPR, we know it's relatively simple from molecular point of view. And when you start the CRISPR project, from your own lab, right, you can have several ways to do this. The first way is, if your lab has enough experience to design and testing the CRISPR reagent, you can do it in your own lab. I do receive some reagent from individual lab because they have experience, they have done that before. The second one is if you don't have a lot of experience, you don't have time, you don't have the hand, you want to do this, you can use other helps. One of the help is from Genomic Engineering Shared Resource here at the University of Minnesota. Or you can use companies. We have local companies here, any other ways. Now it's what we can do with the CRISPR. Here's some detail I will tell you, probably you are more interested in. If you have any question, you can feel free to interrupt me. So what's good for the CRISPR? I will tell you it's good for some project, maybe not as mature as you hope for other projects. For the small indel, or we say for traditional gene knockout, very simple gene knockout, or you want to make point mutation. That's new. In the past, make point mutation can be tricky, but with CRISPR, now it works very well. So I will tell you, if you want to make a simple gene knockout, you want to make some point mutation, you change one base pair, two base pair, or few base pair, and that's a very mature with CRISPR approach. More people, I believe, nowadays more interested in the conditioning allele. That part is still tricky, let's say this way. If you know how to design the CRISPR reagent, and if your donor as a template for your change is around, say, few hundred base pair or maybe less than 2 kb, let's say, with CRISPR approach, it's very mature or relatively mature, let's say this way. You will likely get your mutations. This is, let's say, at the original CRISPR come out. At the very beginning, people very excited because a paper published, they create conditional allele very efficiently. It turns out many labs cannot repeat that result. So there's some confusion, and later it's very clear the original approach to make conditional allele did not work well, at least. But later people when they design the template, provide the donor, they're using single strand or long single strand DNA you can synthesize. And that improved the result significantly. So if your donor less than 2 kb, that's a lot better. How about people try to do as long as 4 kb? In most cases work, but occasionally it did not work. If it's bigger than 4 kb, then the result is, let's say, the efficiency to create your mutation will much less. You can still get it. Actually, we do have, we did some project, and we create the mutations to knock in a fragment bigger than 4 kb. But we also have some cases failed. Now, this is consistent with other institutions, transgenic facilities. When we make a large knock-in, we usually use actually double-strand DNA. There are several different ways nowadays. And the best way, I think, is we will have an initial meeting, me, and also I collaborated with the Genome Engineering Shared Resource. And there's also a coordinator, Brian. And then also the lab, we have an initial meeting to discuss the project, and everybody understand, what the lab want, and how the Genome Engineering Shared Resource, how they plan to design their reagent. And for my part, when I can start probably injecting your reagent, and then the lab has some general idea how long it may take, and or if you have any special requirement for your project, we can talk in the initial meeting, so everybody understand each other. That's probably the current status. I need to explain a little bit more about the, when you make large knock-in. If you read the paper, there are several papers. They all claim they're very efficient method to knock in, say, fragment larger than 5 kb or 4 kb. And I'm not sure exactly what's the problem, but when all the other transgenic facilities want to do the similar project, sometimes work, sometimes not. So it's not, as the paper claim, that efficient, not 100% or whatever. So that's hopefully people here can understand that. The second issue is regarding the when you're creating mutant mice using CRISPR approach, is you may need to put more effort to characterize the founder, because when you use the CRISPR approach, you may introduce some point mutations you cannot tell by simply PCR to genotype them. So initially, you can use the PCR to screening, all the mice you got. But once you had some potential, you do need to confirm by sequence. So that's another issue. So here's a workflow I copied from the Genome Engineering Shared Resource about how long it may take and what's the cost. So hopefully first we can have a meeting with our client, and then from the Genome Engineering Shared Resource, they will design your CRISPR reagent, and they will testing it too, testing a cell line in the cell line. That's required nowadays to test your CRISPR reagent in the cell line. Make sure you design RNA, gRNA, the guide RNA can cut. Usually at this point, seems to me the cutting of the DNA to make the mutation very efficient. The problem is the donor, the template, can be efficient or not. That's a issue. So that's take days in the genome engineering shared resource to make this happen, and maybe 1 week, and then we at the MGL will make the mice. From my point of view, I will look at my schedule when I can start your project. That's actually very dynamic because my situation change, and maybe some of the project I usually plan way ahead of it, because I need to order mice. So these take time. And once I fixed a date, I usually follow that schedule. And I know, say, when this one of the project reagent is almost ready, and then I can schedule that. So usually it's first come, first serve. After I, say, inject your CRISPR reagent to the embryo, we call zygote, then I transfer into the mice. Let's take about 19 days gestation. Then you get a pups. And then I need to wait until mice is 21 days older, I can wean the mice. So that's entire procedure take more than 1 month, So between 1 month or 2 months, you may be able to get mice. And then I will transfer the mice to your lab. And that's, take time for you to characterize mice. So the mice will take a much longer time than just molecular biology. So there's some new approach in the CRISPR method. One is, say, the use adenovirus, AAV virus, to provide template when you want to make a knock-in mutations. Let's say you want to tag a mice with color, with fluorescent proteins, or many other knock-in ideas you have. You can use the rAAV, the engineered recombinant AAV, to provide the template. So there's a limitation for this virus. The limitation will be around 4 kb. So this is actually a single-strand DNA. So as I mentioned, people also found if you synthesize a single-strand DNA, you can just go to some company now, can synthesize the DNA for you. It's very quick, That also improves the homologous recombination, and that's improve your chance to get the mutations. So this is I did one such project successfully in the lab, in the mouse genetic lab. Plan to do more. The other new approach, the people doing the injection in the 2-cell embryo instead of the 1 cell. So the 1 cell we call the zygote, right? That's in most cases I inject CRISPR into the zygote. But there's another approach nowadays, people to inject 2-cell embryo. The 2-cell embryo will improve the homologous recombination rate. That's improve the chance you get the mutation mice. I got the equipment, but I need to try more to make this work. So far, it's not working yet, but I'm going to work on it. Hopefully, it can work. There's some other new approach. It works, but I haven't do it, and actually no plan to do it sooner. That's the last one, this one. That's you can electroporate your CRISPR reagent into the zygote directly in the mice oviduct. That's where they host mouse embryo. So that way, you don't even need to isolate the zygote, and then you do the injection, and then you transfer the zygote back to the mice. So that entire procedure, you bypass it. So it's very good, very excited. Maybe one day you can do it in your own lab, there's some trick probably. That's what I heard, The limitation for this approach is it works for indel to making very simple knockout, or you want to make a point mutation. That's, as I mentioned, that's actually this making simple knockout, or you make point mutation is very mature. So it's also very good if you can use this approach, Yes.
In the CRISPR-Cas9 by adeno-associated, I don't quite understand where the guide RNA is.
Yes, that's a good question. The guide RNA, I didn't point out, is still, you need to separate from this adenovirus. The adenovirus is AAV virus, just provide the template. Yeah.
So with that technology, is the recombinant adeno-associated virus injected, or just infect the embryo? How is that provided?
Yes, I didn't talk about this. That's more technical detail. It's through the infection and the electroporation. So in that case, I don't need to do injection.
Okay.
Yeah.
But you inject the guide RNA?
No, I electroporate it.
Oh, okay.
Yeah. So my understanding is when you do the electroporation, it works very well if the template is small, or if the template is, RNA or single-strand DNA. When you have double-strand DNA, you want to do the electroporation, it can be a problem in not working efficiently.
Do you know if the genome engineering shared resource will provide recombinant AAV vectors?
Yes, at least at this point, they will not do that. But they have other outside company can actually just do this kind of work. If you have some template, you design the mutations, and then you can ask the company to package this into the AAV. Okay. They can do it for you, yeah. And you buy it. Probably not cheap, but I don't know the cost, yeah. When I work with the lab in here, they use outside company to package this our AAV, yeah. So that's the current new approaches for the CRISPR by the CRISPR method, Here's just for your eye only, I think. Let's see the 2-cell injection here. Okay, that's 2-cell injection. Oops. Okay, so here is another display of the workflow, basically the timeline estimate in CRISPR-Cas9. So I didn't mention the traditional DNA injection. Actually, technically it's similar as the CRISPR, just the DNA. That's many people familiar with. That's the original, making transgenic mice. It's similar timeline. First, here I basically say you prepare your reagent, either CRISPR or DNA. Say you can use the genome engineering shared resource companies and your own lab. And after that, I will do the injection. From my part, I will exactly for the injection, I take me about 6 days, let's say, and first by the prepare the mice by hormone injection to the female, and then the final 2 days, I will inject your reagent to the zygote, to the mouse embryo, and transfer back into the surrogate mother. And then you take, 19 days for the mice to be born, another 21 days for the mice to be weaned. When pups is 10 to 14 days old, I will cut the toe. Cut toe will provide material for you to genotype. And at the same time, I basically number the mice. And when you get mice, you can base it on the toe to ID the mice. And hopefully you can get some mutations. And then it's your lab work, to characterize them. So that's about the previous, I just described about the CRISPR or DNA approach. And here's the embryonic stem cell injection to create chimera mice. And that's another way to make mutations. But I can say this request for this type of service is in the decline, let's say in the scientific field, because rise of the CRISPR technology. But it's still useful in many cases. In here at the university, in one lab, basically making humanized ESLs, knock large human fragment into the ESL and make transgenic, make chimera mice, and finally they will, receive basically humanized mice. I think that's what they call it. And that lab, actually, the work going very well. We have many chimeras, and they all want germline, so that's pretty good. And also, as I mentioned in the previous few slides, when you're talking about large fragment, let's say more than 4 kb, maybe 10 kb, you can try the CRISPR, but it's not, successful rate is somewhat problematic, but it works. If you take the embryonic stem cell, it's almost always work. The catch is take time and also expensive in the ESLs. So if you want some conditional knockout allele, you can actually look just for existing ESLs. Almost all the coding sequence, if you look for the ESLs, you can find maybe there's another clone already made for conditional knockout almost any gene, So there's one way you can identify the, say, if existing either mice or ESL already there in somewhere, you can go to findmice.org to find it. Another way is looking at another site is the International Mouse Phenotyping Consortium. There's a website, you can see IMPC. That's I describe as by 19 research institutions to identify the function of protein coding gene in the mouse genome. In that case, you can, looking at their website, you can see if that mice already made, and you can request mice. Or they also have information or link there to see if they have existing ESLs somewhere. And the largest existing conditional gene knockout in the ESL is the European Conditional Mouse Mutagenesis Project. That's basically it was done a few years ago, actually, in a large scale. And I did inject several clones from this consortium, let's say. It works, let's say, in most cases. The problem is for your lab, when you get an ESL, whether it works or not, that's 100% for you. For me, it's statistics, many other labs. For you, it's 100%. So you need to get more than one clone. Just, hopefully you have, say, normally I inject 2 clones for one project. I ask the lab to get at least 2 or 3 clones based on the quality criteria. In this website, the European, the conditional knockout website, they do have quality data. They screen the mice. You can look at the quality data. Based on the quality, you pick up 3 clones to import. When I do the injection, I will inject 2 clones. Hopefully, at least one of them can go germline. So that's where you get the ESLs. And also, you can use companies or maybe some other facility. They provide let's say the gene targeting project in the ESLs. One place can do this, I know, is the University of Michigan, and they can do that for you, because your project is unique, and you cannot find the existing ESLs you design, or you can collaborate with the companies, or maybe like the University of Michigan and design, At this point, we do not provide ESL gene targeting here because the mouse genetic lab is very small, just basically myself. But this can change if you really want to do it here in-house. Maybe you can convince the mouse genome engineering shared resource. They have people. They have people want to work on that. The ESLs. From technology point of view, we can do that in here. I know how to do it. So we can work on this. So it's still a yes and no question if we can do the ESL in-house. For the ESL project, one important thing you need to understand, your ESL need to go germline in order for you to get your mutation. Otherwise, I can do all these. If the chimera mice cannot go germline, you will not have mutations. For the ESL project timeline, it will take longer time for sure. The ESL work is a lot. It can take a few months. Either do it here or you go outside. It will take time. For me, it's similar as CRISPR injection. It's take about 1 week. And after I transfer injected blastocyst into the surrogate mother, it takes 17 days to get the pups born. Take another 21 days to be weaned and sent to you. In the chimeric mice, I will not take any toe clip or whatever, because I will, based on the coat color, to evaluate if they are chimeral mice, is chimeral mice or not, and only give you chimeral mice. And then you can breed it, then genotype it in your own lab. So that's also take time. And then you made your chimeral mice to a wild type mice to see if your mutation can go germline or not. So that's very critical. And you only know that result in the last step. So that's a risky project. I think the entire project is based on the quality of your ESLs. So that's one important parameter for you to consider, keep in mind. So here's other servers. Most request servers for me is the mouse sperm cryo project. Many people, don't want to keep all the mice on the shelf. For MGL to cryo your mice, I require your 2 males per line. And the male mice, hopefully, I have an age in here, but age limitation is flexible actually. But I do like I require your male is proven fertile. I received few mice that are simply too young, the male. It's probably 8 weeks old, but never mated with female. And when I looking at the epididymis, they are very thin and pale, don't have many live sperm around. In that case, when you try to recover the your mutant line, can be a problem. So hopefully, your male also is healthy. People has cryoed male as old as almost, let's say, 2 years, and it worked. But also, you will see receive sperm somewhere they're damaged in the handling the process or whatever, or maybe the sperm cryoed many years ago, and the procedure is different from nowadays. In the past, I don't know, 10 years, 5 years, there's some improvement in the cryo procedure. If your sperm made 20 years ago, it may work, may not. So it's you have to try it, have to do it. Let's say you will know. For my part, I will get 2 male. I will get the sperm out, mix them with a so-called cryoprotectant media, mix them up, and then I aliquot into 16 vials. In reality, it's 17 vials. I always take 1 vial after cryo procedure. I recover them, just culture them, and then look them under microscope, see if they swim. If they swim, then I assuming they should be okay. Because if I really do the IVF to recover them, that's too costly, too expensive. So I don't do that. Just looking see if they swim, and also looking at the density. That's a qualitative assay, and sometimes it's not objective; it's more subjective. Yeah. So IVF nowadays is pretty mature. But again, it's related with the sperm, where your sperm come from. So it's still not guaranteed it will work for your project. Usually we use, either fresh sperm, or most cases it's the frozen sperm. And the egg, I will order wild type female, and then super ovulate them to provide the egg. You mix sperm and egg, you get mice. So that's about sperm cryo. And for the cost, at this point, it's 1, 2, or 3 lines. We always just charge $505. If you get 4 clones, 4 lines, let's say, I will double the 505. That will be 1,010. So that's how we charge. Another one is I mentioned a few times, especially on the website. If you are a cancer center member, your cost will be half. I would say, from my point of view, or from my end, I will charge everybody the same. But the accountants on the back end, when they really apply to your grant, they will see if you are a cancer center member, and then they will discount half to you. So that's how they do the discount. So here's some special service. One is I sometimes do the consulting of the mouse project. One lab, let's say, they want to do 2-cell isolation. Originally, it's the postdoc want to do it, and later postdoc don't want to do it for whatever reason. Asked me to do it, so I did it. We also have a project with some mutations. They want to rescue it, correct the mutations, or make second mutations. So we can do that. That's basically a customized mutant mouse project. I also injected the human-induced iPSC to blastocysts to see if the human cell line can incorporate into the mouse. or we can knock out a gene and then inject. Let's basically use the CRISPR approach at one cell stage. We knock out a gene and culture the one cell stage to blastocyst. At blastocyst stage, we inject some IPS, see if can rescue the mutations. In the past, I tried some porcine pig zygote injection, but so far no people asked after that lab, so we didn't do it because the pig is a much bigger project that take longer time and involve some other labs, I think. There's some other requirements. So one lab asked me to help see, they try to do some guinea pig blastocysts, and they has some difficulty to find it. So I go to their lab and help them to find the blastocysts. Actually, this one is free. I didn't charge anything. So that's a special project. So if you have some, let's say the very unique mouse project is not fall into the CRISPR, ESLs, transgenic mice, you can inquiry, see if I can help or the mouse genetic can help. Yeah, that's the end of my basically explain how the mouse genetic can work for you. If you have any questions, feel free to ask.
Thank you.
So thanks you, and I wanted to congratulate you and Tim also just on the great job you're doing in keeping the costs so low for us, which is awesome. The general question I had is that a lot of people probably want to make conditional knockout allele, a floxed allele.
Yeah.
In your experience, assuming one isn't available as mice or ESL, and they don't want to use a gene trap type of arrangement,
right?
And they want to make it using CRISPR-Cas in embryos, what's your general advice at this point and the experience you guys have doing that?
That's a very tough question. That's based on very specific about your project. Let's say if you want a flux, a very short sequence, say you want flux 1 kb fragment or maybe less, I will definitely say try the CRISPR. It works efficiently. When you go up, because your specific situation, you have to flux, let's say 3 kb. Then that's I will say we can still try the CRISPR approach, but you have to understand it may not working, and we can try again. Second time, the way we charged, that's a issue now. We still don't. It's hard for MGL to price this. If first time didn't work, do we charge the same at first time? Same, same price. It can be expensive for the lab from that point of view. From MGL point of view, it costs a lot, and we also ask to recover the cost. We want to charge more. But at this point, in most cases, we probably, the second time, we only charge the mouse cost. The mouse cost about probably less than $1,000 or a little over $1,000, I forgot. Yeah. If you have a project you want to knock in, let's say 9 kb, 10 kb, then I would say probably need to avoid the CRISPR approach. We actually tried a 7 kb, and it didn't work. That project, we inject more than three times, and it didn't work.
I think for many flox alleles someone might want to make, the distance between the two LOX-P sites might, could easily be less than a kb, and you could make a decent floxed allele by floxing one or two critical exons. Under a situation like that, if you're going to do it by CRISPR and injection into embryos, is that 2 guide RNAs and a single-stranded DNA template for homology-dependent repair, or what exact approach are you recommending? Or a double-stranded DNA, or RAV, or what's the best?
Yeah, at this point, I will say if your template is around 1 kb or maybe even 2 kb, go to long single-strand DNA. To me, that's better. And also, when I looking at the discussion in the email list, the international, there's a email list talking each transgenic facility worldwide. They talking about their experience, and that's basically the consensus of this community. They say, for the smaller template like 1 kb or maybe 2 kb or less, go to long single-strand DNA as a template. We can do the injection, and when the template is smaller, let's say 500 base pair, 200 base pair, we can actually just do the electroporation. Electroporation worked, and we also tried at MGL. It worked well. The electroporation from mouse genetic lab point of view is very simple. Avoid injection. It's very efficient. But injection is not a, let's say, big deal. We can do the injection, and it works very well. So for 1 kb or maybe 2 kb or less, go long single-strand DNA, you still need to design the guide RNA. Usually guide RNA very efficient. When we're looking at the project we have, even smaller template, smaller knock-in or large knock-in, say 9 kb, whatever. Usually it's not, there's no cut. There's a cut from mice when we ask lab to give us data. From that, I depend on the lab. Lab has to give me data so I can talk. Otherwise, I cannot say anything. The lab give me data. So far, we see that it always has a cut. So you had a indel small deletion mice there. Basically, it's knockout mice. But if the knock-in fragment is too big, there's no homologous recombination. So that's also a reason I try to establish 2-cell injection. The 2-cell injection will increase the homologous recombination rate. Hopefully, this can be some improvement if I can do the 2-cell injection.
Related to that question, in terms of knock-in donor, how much of that length of the donor is homologous arms for the recombination? Like, what is the length of that normally people use?
Yeah, actually, it's very short for the arm homologous region. In the 2 end. I think it's I forgot the number. It's probably 50 base pair to 80 base pair at the end. You don't need a large homologous arm for this type of knock-in. Yeah. But for the large fragment, let's say 4 kb knockout and knock-in at ROSA 26, we tried several. That usually we do by so-called TILD approach. That's basically double-strand DNA with only homologous arm at both ends. no plasmid backbone. We have some success, but also we have cases that did not work.
When do you think the prime editor technology will be ready for use in the MGL? Or the
Yeah, that's many people probably interested in this technology, the prime editing. For the MGL, we do service, and we don't do the exploration of a new technology. So once this used by other facilities, and we can try it.
So what will be the rate limiting step here? Will it be other animal facilities getting this up and running, or will it be the genome engineering shared resource here making that available as a service that can then streamline into the MGL?
As an official service we provide, I will wait until other facility can do this. But if the lab willing to take the risk, say even it's not working, you understand the risk, we can do it, because we have technology, and you are willing to do this, why not? We can do it actually, because the procedure basically is the same, but you also need to understand the reagent. For current CRISPR reagent, you can get it from commercial company, For the prime editing, you may need some special way to prepare the CRISPR reagent. So if you can do that, it's fine for us. We can do the injection. Not a problem.
Other questions or comments? So if they want to use your service, the first step is an email to you?
Yes.
Okay.
Send email. I will encourage you go to our MGL website, and there's a link to me, to Tim, and also to the genome engineering shared resource. And we can start from there.
Great. Thank you very much.
Thank you.